Hi, welcome to Biology classes with Lakshmi Chandra. In today's session, we'll learn about a new concept regarding the regulation of gene expression, that is Lac Operon or lactose operon. Before going directly into the topic, let me tell you the importance of learning or discussing this particular topic. We all know that in our cell, in each and every cell of our body, the content of the DNA is same. Let it be the cell of our skin or that of our bones or that of our nerves, whatever it is, the DNA content is the same. The gene is the same. But the expression of gene is not the same everywhere. For example, the enzyme that is produced inside your mouth is not produced in your stomach. The enzyme that is produced in your stomach is not produced by the pancreas or the small intestine. It is different, isn't it? But as I told you before, the DNA content is same everywhere, the gene is same everywhere. But why there is such a difference of production? It is because the genes that are present, they are expressed in a different manner in different parts of our body or in different cells of our body. That means gene expression is regulated in our body. So that is the importance of learning it. We should know how this expression is getting regulated. Only then, if there are some diseases related to this gene expression, we can rectify it. We can find medicines for that. We can find remedies for it. So, as part of the gene expression or regulation of gene expression, today I am going to take a very base topic on that regarding gene expression in prokaryotes, that is in E. coli, how it happens. So, over to today's topic, that is Lac Operon. Lac Operon. So as I've told you, gene is not expressed everywhere equally. It is different in different parts of our body, and also it depends upon three different factors. So gene expression depends on three different factors. That is what we have to learn first. Gene expression depends on, first is your metabolic processes. Metabolic processes, that is anabolism, catabolism, related to the factors that are building up in your body or that is broken down in your body. So that is metabolism. So regarding metabolic behavior, second one, it depends upon your physiology. That is the nature of state of your body, what your physiology is. And third one, it depends upon the environmental condition. It depends upon the environmental condition. So there are three different factors on which the gene expression is regulated. So that is very important for us to know about the three factors. Now here in today's session, I'll take this particular factor, that is metabolic factor, how it is expressing or how it is regulating the gene expression. In order to understand that, let me just tell you the metabolic process that I am taking today is conversion of lactose to glucose and galactose. This is the metabolic process that I am going to deal with today. Conversion of lactose to glucose and galactose. You know, lactose is a disaccharide. It is a disaccharide. That means it is a sugar made up of two monosaccharides. So if this lactose has to be digested, means this disaccharide has to be broken down into its respective monomeric units or into its respective monosaccharides. So those respective monosaccharides are, one is glucose, another one is galactose. Let me just relate to in our case. See, we suppose we have taken it today morning chapatis or idlis or dosha or whatever it is, we have taken that. So it is rich in carbohydrate. So that is a polysaccharide. Now it is undergoing digestion inside our body, and at last when it comes to our small intestine, it is getting broken down into glucose. So glucose is that monomeric unit. That is a metabolic process taking place in our body. Same way, this is a metabolic process in which lactose is getting converted to glucose and galactose. And in order to happen this, there are 3 enzymes which are required. Okay, those 3 enzymes are beta galactosidase. Next enzyme is Permease, and third enzyme is Transacetylase. So there are 3 enzymes. These 3 enzymes are needed for the conversion of this lactose into glucose and galactose. That means if you have a cell and inside this cell lactose is present, and if this lactose has to be converted to glucose and galactose, this one cell has to produce these 3 enzymes. That means it has to express its gene or its DNA so that it is producing these 3 enzymes. So that is gene expression. When does this happen? Only when lactose enters. That is the first one, the metabolic process that I told you. Only if lactose is entering, only then the cell has to produce all the 3 enzymes. Instead of lactose, if into this cell glucose is entering, in this case, does the cell require these enzymes? No, because these enzymes are required only for the conversion of lactose to glucose and galactose. Otherwise, this is not required. So, gene is getting expressed, or gene is producing these 3 enzymes. Gene is getting expressed means gene is producing its product. That is the meaning. Okay, the product can be another RNA or it can be any proteins. We all know that almost all enzymes are proteins. So in this case, this gene is getting expressed or is producing these enzymes only if lactose is present. Otherwise, the gene need not express or produce these enzymes. So if lactose is present, we say the gene is getting expressed or the gene is getting switched on. If lactose is absent, the genes for these enzymes is not expressed or it is getting switched off. So this switching on and switching off of the gene is gene regulation. Now let us see in detail how this happens in the case of a very small cell called as E. coli. In E. coli, how this is getting regulated was actually found out by 2 scientists, Francis Jacob and Jacques Monod. So Jacob and Monod, they were the scientists who first found out how gene expression is getting regulated. And they found out a regulation system that is transcriptionally regulated. Transcriptionally regulated. I hope if you have seen my previous videos, that I have explained in detail regarding transcription in prokaryotes as well as in eukaryotes. Similarly, translation in prokaryotes as well as in eukaryotes. And it will be very, very useful for you if you refer those videos so that you have a base understanding about transcription, and then you will understand today's topic in a much better manner. So if time permits, subscribe my channel and watch transcription, those videos, transcription in prokaryotes and transcription in eukaryotes. Coming back, so it was Jacob and Monod who had elucidated this particular regulation system, and it is transcriptionally regulated. And they made this concept, they gave the concept as the concept of Lac Operon. So here lac refers to lactose, as I have told you. This is all dealing with the metabolism of that disaccharide sugar called as lactose. Now this is a new term, operon. Operon refers to a kind of gene wherein you have many genes attached one after the other, the structural genes. The genes are getting expressed, they are called as the structural genes. And all these structural genes have got just one regulator gene and one promoter gene. So there are many genes which are regulated by one promoter and one regulator gene. Such a structure or a chain of genes, you call them as the operon. This is mostly seen in prokaryotes only. In the case of eukaryotes, for every gene there is a promoter and there is a regulator for regulating its expression. But in prokaryotes, for many genes together, there will be one promoter and one regulator. So such an arrangement of genes, you call them as operon. So we can say that operon is a polycistronic structural gene regulated by a common promoter and regulatory gene. So regarding promoter and all, if you go through transcription video, you will understand what is the importance of promoter in transcription. Yeah, coming back. So Lac Operon, as I've told you, you have many structural genes arranged. Let me just tell you about those structural genes. So here I am drawing two parallel lines. It is depicting the DNA. Okay, it is depicting the DNA. And I am just drawing here three structural genes. Okay, this is my Z gene, Y gene and A gene. Z, Y, A gene. These are the three structural genes in this Lac Operon, and they produce one mRNA. And from this mRNA, you have the Z gene which is producing beta galactosidase. You have the Y gene which is producing the enzyme Permease. And you have the A gene which is producing the enzyme Transacetylase. So only if all these three genes are getting expressed, lactose will be converted to glucose and galactose. Now as I have told you, these three genes have got a common promoter as well as a regulatory sequence. So that sequence I am writing here. Here is the promoter and here is the regulatory gene. The regulatory gene is denoted by a letter I. Okay, so I gene is the regulatory gene here. Now here there is a space that I have left here. Here is an operator gene. Just adjacent to promoter there will be an operator gene which is also equally responsible for regulating the expression of these structural genes. So this is how an operon looks like. So in an operon you can see you have many structural genes. Here there are three structural genes and they are regulated by a common regulator and a promoter. Apart from promoter in this Lac Operon there is also an operator gene. Okay. Now this is how an operon looks like. Now let me just tell you how this is getting switched on and getting switched off. So I hope till here it is very clear to you. If it is getting switched on, means this mRNA has to be produced and this mRNA has to produce all the three enzymes. Now going back to our transcription, from DNA, this is, as I have told you, this is DNA. From DNA, if an mRNA is getting produced, This step is called as transcription. Transcription. And only if mRNA is getting produced, it can produce all these 3 proteins. They are enzymes, and almost all enzymes are proteins. This stage you call it as translation. So gene is getting expressed means here gene is producing its mRNA and mRNA is producing the corresponding proteins. So if I want to stop this gene expression, I can stop it here in the transcription level, or sometimes if I couldn't stop at the transcription level, I can even stop it at the translational level. So gene expression can be regulated in the transcription level or in the translational level. Coming to translational level, if it is in eukaryotes, it can be stopped at that splicing, capping, tailing, all those processes now we can regulate. So this splicing, capping, tailing and all, please refer to translation videos so that you will understand it in a better way. So gene regulation means gene is expressing its mRNA and from the mRNA it is producing the corresponding proteins. I can regulate it by either permitting the DNA to produce the mRNA and protein. That means I am making the gene switched on, or I can stop it. I can stop it at the mRNA level, that is at the transcription level, or at the translational level. So here Jacob and Monod had elucidated the regulation system at the transcriptional level. So that means our Lac Operon is regulated at the transcriptional level. Now how it is getting regulated at the transcriptional level, how it is getting switched on and switched off will be dealt with in the next video. Thank you for now.